a previous video, I covered this paper that seemed to show evidence that when you inject concentrated pig plasma into rats, it caused rejuvenation. However, this research was done in a very small sample of rats, and there isn't much information besides that given. This led Nicolás and Nina, my guests today, to create the Rejuvenation Science Institute, a non-profit organisation to try and reproduce these results. Here we discuss what they've achieved so far and why this research is so intriguing. I hope you enjoy. I brought you on today because we all have a shared interest in longevity, and I know currently you're heading up the Rejuvenation Science Institute in Brazil, and you're trying to repeat some experiments involving injecting pig plasma into rats to see if it can extend their lifespan and other health markers. But I think before we get to that experiment and what exactly you are doing, I'm kind of interested to hear the backstory as to how this all began. Where did this interest come from, and how did you end up in this position sort of right now?
Yeah, well, I think it started basically with Nicolás because he saw the preprint by Harold Kutcher and Steve Horvath and other scientists that they published in 2020, right? About injecting what they call it plasma fraction from pigs into rats, and they observed like 54% of epigenetic rejuvenation measured by Steve Horvath. And so basically Nicolás has a background in journalism, so when he saw this he was like, no, I need to investigate further, I need to know better.
No, my reaction was, well, all the time we see in the rejuvenation space super results, mega good, and okay, so I started to see the details, and I saw the article was the first author of the article was Steve Horvath. So I already knew the reputation of Steve Horvath, and I thought Steve would never put his name as first article in first author in an article clearly a scam. So because of that, I knew it wasn't clearly a scam. So okay, wow, let's see. Then I read what Josh Mitteldorf wrote in his blog about Harold Kutcher, and I said, well, this is probably not true because it's too good to be true. So I wrote to Aubrey de Grey, and I said, Aubrey, did you see that? What do you think? And I was expecting an answer like, yes, Nicolás, you don't have to give much attention to this because it's another, I don't know, non-serious thing. But on the contrary, Aubrey wrote me an email very intriguing, saying, well, first, it's too good to be true. Well, that is not an argument. And he said, well, when the experiment was done by someone in India with not too much supervision, Aubrey gave me some arguments that weren't like, no, this is not a serious thing. Aubrey didn't support it, but he didn't said something bad. So I was very surprised by that. So I thought, okay, I need to continue investigating this. This is worth to investigate because it's too good to leave it there. So I continued. I wrote to Rodolfo Goya, which is an Argentinian who is one of the authors of the article, and he explained me many things, and I thought it was a real thing. And then I decided to write to Harold, and I started to talk to Harold. And what I'm trying to say is that since more than 5 years ago, I am in the same process of trying to know if it's true or not. And in this process, I thought that initially I thought that researching 5 minutes about this, I would reach the conclusion that was not serious. But 5 years have passed, and I still don't know. And I knew Harold, we edited his book, we published his book, we made a consultancy communication services for his company. We made like the institute. We repeated his experiment in the sense of isolating the extracellular particles. And Aubrey de Grey, by the way, told me that probably the rats would die, the young rats would die from an immunogenic reaction when we injected the catcher's preparation, the extracellular particles, and they lived okay. They didn't show any toxicity or in the organs.
In an acute point of view, not like in a chronic treatment.
Aubrey was very surprised about that. Until people in this Harold Catcher thing, they divide like in two believing or two skeptical. Oh, Harold is fantastic, or no, I don't want to hear nothing about Harold Catcher. But we don't have any of these two options. It's the third option. Like, we still don't know if it's true. There are many hints that it's true.
Yeah, not only based on Harold's work, but only other scientists are doing similar research. Like there's a researcher from Spain who injected human small extracellular vesicles derived from human plasma into old rats. They had like a very good response. There was less fibrosis in the heart and several other organs as well. So there are other researchers doing similar things that help to confirm as well Harold's results. The thing is that Harold is the most impressive in terms of what they report as epigenetic rejuvenation. Is the biggest amount of epigenetic rejuvenation. And some years later, I think it was in 2023, right? That they published a peer-reviewed article. And now Harvard, using a different method, they reported 6 to 7% of epigenetic rejuvenation on average, because in some organs it was more than that. So basically what Nicolás is saying, like, we want to know the truth, is not that we're saying that they are doing something nefarious, doing something like that. It's just like, can we like reproduce this? Is this true in this sense, in the scientific sense of someone does an experiment, it needs to be reproduced by other groups or other people, like as we were talking the other day with our collaborator, because we're doing this in collaboration with a university here in Brazil, with a professor from the Institute of Biology. And he was talking about, for example, the rats, that even if you give the same kind of feeding to the rats based on the macronutrients, he was saying that the United States, they use lard as a base for fats. And here in Brazil, they use oils from plants, so even that can change like the result, because the rats have a different feeding. And so yeah, that's it. We're trying to see if it's true in that sense, because a lot of people from this field want to have some kind of therapy to rejuvenate people, to increase longevity. And this is one that is very impressive if it's true, if it can be reproducible.
And there is a problem that the original group took 3.5 years to say what they gave to the rats, like to publish the article in Journal Science. So only then, in October 2023, it was possible for anyone to reproduce that experiment. And so, I don't know, maybe there are people out there reproducing the experiment in their companies or their labs, but I see that there is first, we can't wait for people to do it. I hope tomorrow someone publishes an article saying they reproduce it. Fine, we don't need to do it, but we can't wait. People are dying now. So, and another reason to do it is that I see that the incentives regarding this research can create problems for scientists to reproduce it. First, because it was already published, so the patent was already requested. So whoever reproduces can't request a patent about it. So investors can have difficulty to invest in this because they can't have intellectual property. And on the other hand, the academics, the scientists, they don't like to reproduce experiments. They want to make something.
Original research.
Yeah, on original research, change something. But this result was so good that, no, we have to do the same experiment and see if it's reproducible. So I think the academic world and the investment world, the companies world, they probably have problems to do it. And so that's why we founded this non-profit research institute here in Brazil, because we wanted to tell everything immediately. If we see that the rats rejuvenate, we want to publish methods, materials, results, very detailed, in order people can do it.
But we also want to publish even if the results are bad. So I don't know if many scientific publications will accept, I think, negative results, usually, I think that's how they say, but we're trying to publish it or at least to talk about this information. We have like our website, our newsletter, we want everybody to know the result.
Yeah, and because it's a non-profit, we started this crowdfunding campaign. So this experiment cost $75,000, and we already raised it $53,500 is $70,000. It's 72% of the total is $53,500. We had some big donations. One from DoNotAge.org that donated $25,000. Hillis, it's an NGO from Belgium, donated $10,000, and we had also like 30 donors or 40 donors of smaller values, smaller amounts. And when we finish this experiment, we want to let the rats live too, because in the original experiment they killed the rats after 5 months. Wow, it's true that the COVID quarantine started in India, and maybe they had to shut down everything, or maybe they didn't expect that awesome epigenetic results. but they killed the rats, and we want to let the rats live because we can say, oh, the rats are rejuvenated 67% epigenetically. For scientists, that's great, but for the general public, it's almost nothing. But rats living indefinitely, everybody can understand that. A 70 years old person can say, oh, I'm old, but I am very strong and I'm very good. But they won't reach 150 years old. Humans don't reach 150 years old. So if we can make a rat live the equivalent, like pass the maximum lifespan of rats, that we can say, oh, this is the world record of rats living. So, and we double that record. I don't know, but what I'm trying to say is that lifespan experiments are very clear in the results that something was affected in the aging process. So we will start this longevity experiment at least for 12 months.
If we see positive results in the original experimenting. So if the rats either are apparently having younger phenotype, or if they are at least like non-aging, something like that, So if we have like positive results, we want to keep the rats alive and do a longevity experiment.
And simultaneously to the longevity experiment, we want to make a good laboratory practice test in Brazil in a facility specialized in safety tests for rats, in order to see if the PEPS, PEPS is the acronym to Pig's particle extracellular, pig plasma extracellular particles, and then we will see if the PEPs are safe according to the regulators of Brazil, which the agency that regulates tests in Brazil follows the FDA and the European Agency of Regulation. So we will see if it's safe, and if it's safe in this safety trial in 2027, we want to make a human trial in 2028. In Brazil, if we want to make a human trial, you need two species tested before. But if you have one species tested, like rats, you can use the therapy in compassive patients, people who has a condition that is killing them, and they already try everything, nothing worked, and they don't have options, so they can try something that was tested in one species, if it was correctly tested.
Yeah, if it's proven that it's safe in that species.
Safe and effective in rats. So we want to make that test in 2028. And, well, yes.
Yeah, we're talking too much.
That's great. I don't have to do too much. No, it's fascinating what you guys are doing. And as you said, you've already got donations, which I think just supports the scientific backing and interest in trying to reproduce this results. And so I guess before we go more in detail into how exactly you're isolating this fraction and then injecting it back, I guess, what is the sort of scientific rationale for doing this in the first place? Like, why was Harold so interested in doing this experiment? And I think, of course, maybe my viewers are familiar with heterochronic parabiosis and the sort of famous experiments, but could you perhaps explain a bit about the science, or why do you think that this approach would rejuvenate the rats?
Yeah, I think this approach is based on the idea that we have signaling in our body that basically can tell us that we are old or that we are young. And this comes from first of all, this heterochronic parabiosis experiments in which two animals, usually rats because they're bigger, their circulatory systems were connected. So two different animals, and one was young and the other one was old. And so the scientists would keep these animals connected, their blood streams connected, and then separate them. And they would observe that the young animal kind of aged more rapidly than expected, and the older animal rejuvenated. So there were analyses. I think that the first thing that they noticed is that when they killed the rat and analyzed the organs, they showed less yellowing in several tissues. That was a sign of aging. But then there was a lot of other experiments, because this began like 100 years ago, something like that. They started doing this kind of experiments. But even today, some groups do that, but there are several biological markers that they check, and they see that the old animal appears to be younger, or to be healthier, to show a younger phenotype. So Harold, I think he was based on that, and also on the Comboys, the Irina and Michael Comboy. They worked a lot with also plasma dilution. So they base the idea that we have like pro-aging factors in our blood, and if we dilute our blood, we kind of rejuvenate, or at least age, decrease the velocity of aging. So I think Harold kind of was based on that, and he wanted to do like a plasma exchange between a young rat and an old rat. That was something that he wanted to do, but he didn't have like an equipment to do it, because it's very rare. I think there is like one in Germany, something like that would do something like that. Because in humans that we have like the therapeutic plasma exchange that some people have to do in hospitals for several health problems. So he wanted to do that in rats in an heterochronic way, a young rat and an old rat. But he couldn't do that, and so he said, well, maybe I can isolate from the blood the thing that I think is something that is a pro-youth factor, something that promotes youth in an old animal, for example. So he decided to take it from a bigger animal, because he wanted to concentrate it to give to a small animal to have like a stronger result. That was basically the reasoning that he explained in the book that we published.
In fact, in the book, The Illusion of Knowledge, he has a theory of aging, a very beautiful theory of aging, because we see in the longevity or rejuvenation space, people comes up with a substance. I don't know. This substance we tried in rats, and he extended in 20% the life, or and people try things without a clear theoretical reason for that. And what I like in Harold's experiment is that not only he has the most successful experiment of rejuvenation of mammals, but also the most compelling and complete theory of aging that is explained in his book. He is a biologist. He goes to the beginning of life to explain that aging started with sexual reproduction. So bacteria don't age. They reproduce asexually. They are the same that their parents, So Harold has a theory that started there, and he analyzes the unicellular organisms to show that the way the unicellular organisms age, depending on being asexually or sexually reproduced, are similar to what cells, human cells, how human cells behave. For example, the Hayflick limit, that after 60 reproductions the cell can't reproduce anymore. He shows that the sets of I think are paramecium or unicellular being, they behave collectively like the cells of humans in terms of the numbers of times they can reproduce asexually, and after that number they have to reproduce sexually. Like it's a rejuvenation, like a rebirth. So and then Harold transported that to so Harold then identified that there should be a mechanism very ancient in life to regulate the lifespan. So he also states that the circadian the circadian rhythm, the day and night, are the clock of this tic-tac. Is the way the cells regulate the reduction of efficiency of the repair mechanisms and the reduction of efficiency of energy production. That Harold say that these two mechanisms regulates the rate of aging. And Harold also says that despite the rate of aging is genetically programmed, it is epigenetically programmed. Epigenetically implemented. It's genetically programmed and epigenetically implemented, but the epigenetic we can change. It's
more easy.
It's already proven that we can. Rejuvenation for cells already exists with the Yamanaka genes. If you use partial cellular partial cellular reprogramming, you can rejuvenate a cell. So we already know that that rejuvenation is already possible in cells. So when he made his experiment with exosomes or small cellular vesicles, that was based in a real biologist thinking about aging, and not just people with a lot of intelligence, but that are not necessarily biologists. So that's what I like in Harold theory. Is theory and practice. It's a theory of aging and the best results got. So we have to confirm it. So we can't escape of that. We have to make science here. People are doing too much. Too much business. Business are important, but we have to have priorities. Business have to exist for having, rejuvenation, and not the opposite. It's not the rejuvenation that have to exist business. It's the opposite. And not anything can be done with business. Basic science is very hard to be done with business, because when you make basic science, the chance of you catch the wrong line is very big in basic science. Like Google spent like $4 billion in Calico trying to make rejuvenation. And if they chose the wrong line, if the owners of Google, they are not biologists, by the way. They are very good in computing, but if they got the wrong the wrong line, because they can say, no, but we have infinite money. We will hire the best aging science in the world and the best rejuvenation science in the world, and they will know. But who will they hire? Who is the scientist that rejuvenated mammals? Or, they hired, I think, Cynthia Kenyon that had, I'm talking about 12 years ago, they hired a person that had rejuvenated nematodes using, okay, fine, but
doing that thing with the Dower estate, right?
Yeah, no, with genetic modification that extended the nematode life in 10 times. Okay, fine, but it's a nematode, and if they chose the wrong line, it's wasted the money, and that happened with the owner of Amazon, the owner of other companies that started rejuvenation companies. And people are expecting, oh, Jeff Bezos will save us because he's very rich. Yeah, but what he know about biology? I mean, he studied biology, he studied aging, he spent 4 or 5 years studying that? Of course not. He will hire someone. And in basic science, the rules are different from in applied science. Like, for example, the plane was invented by, the Anglo-Saxons say that it was the brothers Wright. But here in Brazil, we say that it was Santos Dumont that make a plane and went to France to give around Tour Eiffel with his plane. Okay, but it was at the same time, more or less. The thing is that Santos Dumont or the brothers Wright weren't big companies, weren't the government with a lot of money and capital. No. They were, the great inventions in basic science are not necessarily from the big capital or the big governments of the big powers. Take radioactivity from Marie Curie, for example. She and his husband, they didn't have any money. They didn't have any money to discover radioactivity. They had a small, house in the university to make the experiments, and they discovered radioactivity. After the discovery in basic science, then the great powers of the world, the great capital and the governments, they take this technology and develop. But the basic science thing is different.
Make it available as well for everybody.
Yeah, they more perfectionist. They perfected the technology, but the discovery is like the discovery of vaccines. It was a guy injecting his cow, his son with his cow. Something totally out of the great powers of this world, I'm telling this because why two Brazilians? I'm a journalist, Nina is a scientist, but I'm a journalist. So why we could do something like this? We are not inventing anything. We are reproducing something. Who invented was Harold. We just see everything, and we think, someone have to reproduce this. So we are friends from Marcelo Mori. Marcelo Mori is a world-class aging scientist that we have the luck to live in the same city. Because there is a very big university in our city. So we are friends of him since 2018 because we published Aubrey de Grey's version of Aubrey de Grey's book, the version in Portuguese here in Brazil, and we invited Marcelo. So we saw that we had all the conditions to make this experiment. We just needed, to found the institute, to raise the funds, and to do the job. And we are almost there.
Yeah, I think I really like your point that you're right, that there is no true correlation between amount of funding and backing in industrial settings versus academia and actual success. And so, I'm an academic myself, so I like to think hopefully I can also at some point contribute. But I think, I've read Harold's book and I understood his background and his insights into why he decided to use the plasma from, in this case, the young pigs. But I guess, blood contains many things, and you mentioned exosomes, or these extracellular particles, but there's many things besides that just in plasma. So once you've isolated this plasma from young pigs, what is this processing steps, and what is it that you sort of end up with and concentrate to inject into the old rats?
Yeah, so we first just isolate the plasma by centrifugation, like very simple. Then we add PEG, polyethylene glycol, to precipitate basically the extracellular particles. Then we centrifuge again just to isolate the precipitate, and we dilute that again and do pass it through a size exclusion chromatography. So just to get the smaller particles. But it's not like and then we just do a dialysis to concentrate the solution. So it's not like a procedure that is very like isolating something very specific. Harold himself didn't do many analysis in terms of characterization, so he just did like a Western blot to confirm that there are exosomes in his preparation. And also I think he did like something to measure the sizes of the particles, just to check the sizes. And when we did like our first experiment, because we did like a smaller experiment just to check if this preparation could cause an acute toxic or an immune response if injected in the same amount, because Harold injects in the rats a very big amount of this. So in this first experiment, we didn't do any kind of characterization. We just like followed this protocol, and the rats were fine. They gained weight. We did a histological analysis, and the organs didn't show any sign of toxic or immune response in this acute kind of way. And so now in this next experiment, we will we will inject in old rats to see if they rejuvenate. But in terms of characterization, we still are not doing that in this first step. But if the rats show positive results, we, besides doing a longevity experiment, we also want to do characterization. And this is something that Professor Marcelo Mori, our collaborator, he's very specialized in that. He works with exosomes, small cellular vesicles. So that is something that we want to do. We want to do like a more analysis to see exactly what is there, because Harold, I think he mentioned it, that maybe it's not only the small cellular vesicles, but other small cellular particles that may contribute as well, like ectosomes,
supermeres, exomeres.
Yeah, he was talking also about other things. I think in interviews he talked about that. So the protocol is very simple. In our article that was recently published in a peer-reviewed publication, we tried to describe it with as many details as we could, because in Harold's paper they didn't, like, some things were kind of missing, not very clear. So we try to do it very clearly, and so it's very simple. It's not like it's kind of a classic way to extract small extracellular particles or small extracellular vesicles.
Yeah, what is special in what Harold did is that he injected a lot. If you see the scale of how many small extracellular vesicles he injected, is much more than what people are injecting. And also it was cross-species, pigs to rats. So that's important because, first theoretically, because if it's conserved between two different species of mammals like pigs and rats, it reinforces the theory that it's a very ancient mechanism in control of aging. And also, we want to do, during the longevity experiment, this characterization. I mean, when we are letting the rats live, then we will make, Maury always insist on that. He said that we need to make the characterization to publish. We have to check if there are indeed small extracellular vesicles there, and the purity degree too. And, you know, we don't think that if it works, humanity will be rejuvenated using pig's blood or cattle blood. It's not possible. There is not enough animals, and it wouldn't be ethical or practical. But we think that if you can prove that it's possible, even in humans, you know, you could do it in humans with human blood. Theoretically, I don't know, if 50 people of 20 years old donate half a liter of blood, you could make a dose for a human. So you could do it from humans too. But if we can prove that rejuvenation is possible in that way, we expect that we learned then how the cells communicate in order we can synthesize that. You know, there is a code there. I mean, the cells receive some information that makes them, you know, activate or regulate their efficiency of repair or the efficiency of energy production to have a phenotype old or young. They receive an information. So instead of using blood of young animals, they codify that. They codify the language of the cells in order we could synthesize this signaling. I mean, behind Harold's theory, there is an aging by signaling theory. I call it aging by signaling theory, that aging is controlled by signaling. So with the correct signaling, we could control the age phenotype of the cell. So together with Maury, we also want to explore that. But that is much more complicated, and sincerely, we don't have to do everything. We want other labs do. We want to prove that rejuvenation is possible, and then people start to do that kind of experiment. So, but we don't count with that. We can't count on, you know, others' lab. Okay, if they do, excellent.
And we'll help them as well. Like we are talking with other scientists. Sometimes they send emails, or we do meetings with other scientists that want to collaborate, or they want to do a similar experiment, so they ask us questions, you know. So it's not something like we want to do it because we want that to exist, you know. We want that line of research to be explored, you know. Yeah.
Yeah. So we are talking. I sent emails to hundreds of people in the rejuvenation space. I have a list of contacts that I accumulate in 9 years of activity. So I talked to news outlet. I talked to YouTubers, well, like you, right? And YouTubers from Brazil too. But I think that we need to rejuvenate the red. You know what, Eleanor? Despite we are doing this to prove if rejuvenation was invented or not, the person I want to convince first is me. I want to know if rejuvenation is possible in this way, because if it's possible, I will be a different person. I will do things differently. I won't talk to my mother that is 78, 70, well, she will be 78 in October, years old, or I have like 20 relatives almost dying. Uncles, the wife of the uncles, grandparents. So if I know the rejuvenation is possible, if I know, because I don't know, but if I know it's possible, if I see the reds rejuvenated in front of me, I will change. All my life, my activity will change. I will talk to my friends like, hey man, your mother is dying, help us, So I want to know. So, so I want that other people, I invite people, I wrote to the Hillis email list, and I said, okay, I invite everybody to come to Brazil in the moment of the experiment. You can go to the lab with us. We can go to the farm to collect the blood of the pigs. We can see everything, to check if it's true. So, okay, let's see. We already raised much more money than we thought it was possible. I mean, we have a simple life here in Brazil, and but we were relatively surprised to be able to raise 72% up to now. We know that our path is very, long, that there are other experiments to do later, but we are more confident now that, we publish like an article in a website, and people appears, people are interested in this research, and there are many people interested, common people, followers of Rejuvenation Science, and we talked with scientists and entrepreneurs of this field, and we also felt the interest, but the problem is that there are economic, political, regulatory, and even psychologically problems to carry out this experiment. And one thing we wanted also to tell you and your audience is that here in Brazil we are making this alliance with Professor Mori. Professor Mori is he was the representative of all the professors of the university. He's a very active person in terms of academic activities, and he's trying to found the first aging research center of Brazil with funds from Brazilian state, and he has this project, and he asked us to write a letter as an institute supporting his request for Brazilian government, to
show that the community has an interest in that, to show the Brazilian government that it is something worth it to create, because they will give a lot of money for that, right? And also he talked to us you guys know a lot of people from the longevity, the rejuvenation community. Could you get like support letters for us as well, like even from abroad, and from companies or NGOs or researchers, because that is something important. And
yes, this center of research could make collaborations with any company or any NGO or any laboratory in the world, And so he asked us to show not only Brazilian government, is the state of São Paulo's government, is a state from Brazil who is doing that, to show the state that a center of aging research here could improve collaboration with the whole scientific world in the aging and rejuvenation or longevity space. So we are not we are in this until the end. I mean, we want to make rejuvenation, and we are not just scientists. I mean, I'm not even a scientist, but we are like militant organizers, coordinators of something that can rejuvenate people. Earning money, not earning money. I mean, that people are very linked to some production system. Like some people loves the investment system. They want, intellectual property, and they want investment. And some people love, state funds. Oh, the governments of the world have to put billions in rejuvenation research. Okay, but we think that it's not so important the system you get, but the goal. For example, we want to produce information on how to rejuvenate a mammal, and we will only produce information in our institute, so we can do it with donations, because we can tell everything then. We make available for everybody. But if someone want to implement this, like a clinic to inject the small cellular vesicles in someone, it's different. It's a different economic system. You have investment, because it's not the same produce information than produce, a service. So we are doing from that point of view. It's not just science here. We want to move the levers of society in order this can be true, and not only a dream, not only a possibility.
Exactly. And yeah, I think your approach is very unique, as of course, firstly, you're doing a reproduction experiment, a reproducibility experiment, which is unfortunately quite rare still in academic science. And not only that, I think often in aging space, it's more typical that we have an idea of like factor X that we think might rejuvenate. So we give the mice factor X and then see if they rejuvenate. Whereas in many ways, you're working the opposite direction. You're going to try and reproduce data that suggests if we give them concentrated plasma, we already expect rejuvenation. If we can reproduce that, can we then understand what's in that plasma that's causing that effect and work backwards? So I think it's also just from a scientific perspective, like a very interesting approach. And of course, I mean, my interest is what exactly is causing this rejuvenation. And I know that you said that this is not your immediate concern, but something that you're also interested in. And I think just the scientific field and probably collaborators later on would help with that. So yeah, I guess, I'm always interested in the biology, but it's also about the practical funding, and the actually doing the experiments can also be challenging. And so I know that you most recently published your Acute study to check about the toxicity. And I guess, what's next? When is the next set of experiments happening? And when can we find out or see some of the rejuvenation data?
Yeah, so basically to be updated with everything we're doing, we have a newsletter. So in our website, I think you link it here, but it's rejuvenation, rejuvenacimento.com.br. rejuvenate.org. Rejuvenecimento is the word for rejuvenation in Portuguese. So you can subscribe to the newsletter, and we send monthly newsletters to keep everyone updated. But the next step is this rat rejuvenation experiment that we're doing in June of 2026, so next year. Basically because we have to wait for the rats to get old enough because Harold used 25 months old Sprague Dolly rats, and it's very hard to find rats that old. So basically we got them from the university that is our partner in this, but they were 9 months old at the time. So in a way that the experiment is already happening because we are keeping the animals alive, we're taking care of them. Not we, but the people in the university. But we will do this experiment, so in June of 2026. So we will again get blood from pigs. So it's the blood that would be discarded basically during the regular slaughter for meat consumption. So we collect that blood. We do all the experiment in the university. So actually a postdoctoral student will be hired to do that, and then we will inject it in the rats, and we will do it a bit different than Harold in the sense that we will have more rats. So we want to have 10 rats that are 25 months old that will be treated, and then 10 another 10 that are the control rats, same age. And also we will have 10 rats that are 7 months old, because we want to see if these extracellular particles can keep them young, basically, to prevent them from aging. And another 10 animals for control the same age. So in total 40 rats, and Harold did with 6, I think. So just to increase a bit the N. And so we will basically follow this protocol, so the same way he extracted that we already did it, and we will inject it 2 times. So a first dose that is actually divided in 4 injections that are given in alternating days, but a first dose, and then 3 months later, second dose. And we'll basically keep the animals for 5 months, and during these 5 months we will collect samples from blood that will do epigenetic age of the blood before the experiment, and also after the 5 months. And also we'll do another several blood tests, like to check like liver enzymes, kidney enzymes, and cholesterol, and several of these biomarkers, and also memory testing and grip strength testing. So this will be done in several points in these 5 months. And as we said, if the animals show positive results, we want to keep them alive. So we'll not stop at 5 months. We'll want to keep giving them doses every 3 months, the same dose, and keep them for 12 months after those 5 months. So that's the idea to see if they basically to see the lifespan, to see because after 5 months the older rats will be 30 months old, so it's very old. So basically we want to do this experiment, but the crowdfunding that we're doing right now is this $75,000 is for this, just the rejuvenation, not the longevity experiment. But yeah, that is the next step.
The longevity experiment, theoretically, if the rats really rejuvenated 67% epigenetically, they could live indefinitely. I mean, if they're young, why would they die? So
yeah, one thing that I think maybe it's important to mention is that the epigenetic age measurement we'll do with Steve Hurwitz's foundation. So we're trying to do as close as possible as Harold did to try to see if, to try to reproduce it, basically. So,
but theoretically, if the rats can be kept young, they could live indefinitely. I mean, this we can say they can live forever. Well, I don't know. That's a possibility. We have to mention it. It seems sensationalist, but the aim is to be young as long as we want. So if we can do it with rats, the aim is to make do you know that movie Highlander that they were immortal humans that fought, and if they could die if they with a sword they cut the head, but if not, they could live forever young. So we joke that the rats would be Highlander rats. I mean, they could live forever, but if a truck goes through them, they will die, of course. But we have to say it because it's true. Because it's true. It's what we want. And we, in our website, as Nina said, we have a newsletter. We can update everybody who wants. People can write to us if people can make this letter of support of the project of Professor Mori, can write to us and say, okay, I have a company, I have an NGO, I want to make a letter of support. If people want to donate, there are a button in our website. It's possible to donate in every way. And I think...
And all the money donated is going straight to this fund for this $75,000.
Yes, and we want to be open about that. I mean, people can ask us without, yes.
No, sorry. It's just we also, of course, after this study, we want to publish it in a peer-reviewed publication, but that takes longer. So in our newsletter, people will be updating quickly.
Yeah. If someone wants to come to Brazil to see the experiment, they can. It's important enough, it can save the life of their relatives, of their self. We see people spending hundreds of thousands of dollars in so many things. Maybe they can come here to Brazil to see, because that's a fight for life. We are all together in this, and this is a collective effort. I mean, we are not doing this alone, and we need to make science here and to know the truth. That's the point here. I remember in 2020 when I first read the Harold's article, I couldn't sleep at night because,
Because you have insomnia.
No. It's because it was too good to be true. So I was thinking in my head, rejuvenation had been invented, and I'm still 5 years trying to really check it. I'm a journalist, right? So I'm trying to make a journalistic job here. So I check it with several sources, and I had to make the experiment myself. I would like other people do, but sometimes you have to, if you want something done, you have to do yourself.
Exactly. And yeah, maybe in 2 years' time now, we will have some insights as to whether this can rejuvenate the rats. So yeah, I think I'm what you're doing is very important, just like for scientific reproducibility. And if this result turns out to be true, then of course that's really big news for the longevity space and of course just globally, right? So yeah, I can only wish you the best of luck, and I'll of course put your links to your site in the description for my viewers to check out. But yeah, thank you for telling me today all about your background and your motivation for this project. And so yeah, best of luck.
Thank you.
Yeah, and
Thank you so much.
thank you for your activity on the videos. It's awesome. There are no many
Not so many.
not so many YouTubers in the rejuvenation space. So congratulations.
Thank you very much.